Going to go. So we're going to talk a little bit about the basics about how the dentist gets involved in systemic disease. And as we've heard this morning, periodontal disease is expressed by several clinical and radiographic parameters that were more prevalent in people with coronary heart disease. And this has stimulated a lot of interest in the mechanisms that might be common to those two problems. And this is a fairly recent publication, as you'll see here, from the Journal of Clinical Periodontology. And they asked the question, is oxidative stress and inflammation the link? And so what we're going to focus on for the next 45 minutes is the linkage between oxidative stress and inflammation. And for those of you who don't want to hang out, you can just come back for the number later. The bottom line in all this is that glutathione is going to be the link between those two mechanisms, and I'll show you why. That's the molecular representation of glutathione, with that yellow ball being the sulfur group that's the prominent component. Glutathione has some advantages over some of its components, however. It's made of 3 amino acids. You've heard of most of them. Cysteine has been used to build glutathione. And people always ask, why don't we just use cysteine instead of glutathione directly? And you can. It has some advantages, but there's certain situations under oxidative stress where it doesn't get formed into glutathione. Glutathione has some distinct advantages. It's more soluble, and it interacts with enzyme systems, as you'll see in a few minutes, which enhance its function. So here's the cysteine molecule with the sulfur group. They combine to form glutathione, and glutathione has a number of functions we're going to address. Why do we need glutathione? What role does it play? Well, there's back in time when apparently our world was in a different form than it is now. CO2 was prominent in the environment, and blue-green algae eventually developed, which then released oxygen as their byproduct of photosynthesis, and created what's called the Great Oxygen Crisis, somewhere between 1.5 and 2 billion years ago. And so all these organisms that had been living in a CO2 environment suddenly needed to deal with oxygen. And the way that they dealt with it was to incorporate what's called an organelle, a part of the cell that can use oxygen. Now, I'll tell you right now, there's a big controversy potentially brewing. It's probably not going to be in the newspaper for a while. But we used to think that mitochondria were pulled into the cells by this nucleated cell, and somehow they got along. A fellow named Nick Lane, L-A-N-E, has written a book, one on oxygen and then a follow-up on which he calls Sex, Power, and Suicide. It's kind of a catchy title. But that's the story of how mitochondria function in our cells. And he makes the thesis in this book that the mitochondria actually created this extracellular protection and store some of the excess DNA that they didn't need to survive over in what we call the nucleus. So I'll leave you to get into that conundrum of what came first, the nucleated cell or the mitochondria creating a nucleated cell. But the fact is that the mitochondria can use oxygen to make energy. So it's really got cells that use mitochondria are so-called nucleated cells. All nucleated cells have mitochondria. Have an advantage in that they not only use oxygen, but they make energy. So here's our complexes on a membrane, complex 1 through 4, taking the electrons that are brought in there from the food that you eat through the Krebs cycle, for example, and then using the energy from those electrons to activate phosphorus and create ATP. At each step, those electrons are used for that, and then at the end, they've done their function. They're still electrons and still powerful, but with less energy than is needed. And usually then it goes off to oxygen, which is kind of a sink, if you will. Some people refer to that as a place where electrons can be accumulated and turned into, along with hydrogen, into plain water. And that works really efficiently. Nothing wrong with that, except that even in the best of circumstances, 1 to 5% of the oxygen that's being used gets formed into a radical. And that's where the problems start, and that's where most of the free radicals that you hear about in literature are generated. They're right in the mitochondria. And that's fine, except for the fact that something has to be done with them, or they're going to create an oxidative stress. So if you remember back in chemistry, this reaction, Leo, lose electrons and you're oxidized, and Ger, which is gain an electron and you're reduced. Well, this is happening when you have a free radical. It's got this extra electron and it wants to pair up. So it's going to grab a hydrogen, a proton, or something from whatever is around it. And when it does that, if it doesn't pull it from an antioxidant, it's going to pull it from a lipid membrane or a protein. It alters the structure and function of those materials, and that's where free radicals become a problem. Some of them can create enough problem that they cause damage. So this is a quick summary of that, which I think is probably in your handout. There's going to be some extra slides here, so overall most of this is going to be covered. But if you're trying to follow it directly in your handbook, I've got some extra slides in here. So here's the definition of the free radical. And the reactive oxygen species are what is referred to as the free radicals of oxygen. And that includes both the superoxide ion, which is the first one over there, and the second one is the hydroxyl radical. And the hydroxyl looks like water without that extra proton, and that's what it is. But it has got a really powerful action to pull that proton. The superoxide radical can kind of float around and for some distance. The hydroxyl radical does not. And we're going to nobody does. What am I saying? I sent some slides in which I thought were going to be placed in a handout. Oh, that was the last lecture I gave. Thank you. So the hydroxyl radical is the big bullet. That's the one that's going to blow holes in membranes and things like that. So it's got to be dealt with. And if it's not dealt with, it will disrupt the mitochondria and prevent them from functioning properly. And again, here's where these radicals are made primarily. And a material called superoxide dismutase is found in both the cytoplasm and in the mitochondria, which will turn the superoxide radical into hydrogen peroxide. But unfortunately, the hydrogen peroxide can break down further and create hydroxyl radicals. Now, what glutathione does in the mitochondria as well as in the cytoplasm of the cell is work with an enzyme called glutathione peroxidase, which is a selenium-based enzyme that won't work without its specific substrate, which is glutathione. So what happens then is, with glutathione peroxidase working, you can turn peroxide using a couple of glutathiones into oxidized glutathione in water. So this reduces the peroxide radical, which can be broken down by the exposure to transition metals like mercury or even oxidized iron or copper into the hydroxyl radical. So the glutathione peroxidase plays a role in preventing that. And here this is depicted in a graphic form, which illustrates how that proton or hydrogen ion is actually stuck on the radical there and creates harmless water. So glutathione plays a really critical role in the ability of our cells to make energy. And also the other things that happens in the mitochondria, we don't think about this, but there's a large number of functions that occur there, including the enzymes that help break things down, the P450 enzymes. They work inside the mitochondria also. And part of the heme that's formed from the porphyrin biochemistry that you've heard about recently, part of that starts in the mitochondria, then the portion of it is taken care of in the cytoplasm, and then again the heme molecule goes back into the mitochondria to form both these complexes that are used to make energy, as well as the components, the P450 components. And they all have iron in them, and they're all creating these oxidative stress situations. In addition, the superoxide radical can combine with nitric oxide, which is also formed inside the mitochondria. And if there's something not present, then you'll form this molecule called peroxynitrite. And so that's the reactive nitrogen species that you may have heard of that's formed. So both of these, reactive oxygen, reactive nitrogen, create free radicals that can cause damage to lipids, proteins, and even to the DNA, both of the mitochondria, which disrupts mitochondrial function, and eventually these radicals are exported out of the mitochondria and can get into the nucleus and damage the DNA in the nucleus. Now, in the cytoplasm, we do have catalase. That's a material that can take hydrogen peroxide and convert it to water. But the key is, and the reason I focus on the glutathione is that the mitochondria itself does not have catalase. So it's really dependent on glutathione and the peroxidase, glutathione peroxidase. But it's so dependent that the cell has developed a high affinity energy requiring transport for glutathione to bring it into the mitochondria to maintain mitochondrial function. And I think this is the real key to understanding why glutathione has played so many roles in cell function, is that it's very basic to the very function of the cell, the ability to make energy, the ability to create enzyme reactions. It's all dependent. If glutathione peroxidase is not available, then the mitochondria quit working. When your mitochondria don't work, your cells don't work, and that spreads into tissue damage. Also, you can have chlorine available, which creates, through these hydroxyl radicals, can create hypochlorous acid. And neutrophils actually use that in the presence of hydrogen peroxide. That's what they use to damage invaders and break apart debris that the neutrophil and the macrophage has absorbed. And that's kind of the cleanup mechanism. It's like a big street sweeper going along, and they pick something up. Well, if it's an invading organism, they can just zap it with these high oxidative stress materials. And the hypochlorous acid is also removed by interactions with glutathione. So this is kind of a repeat of some of the concepts that we've talked about, emphasizing the fact that the metals, this is where the different metals come into play, but they're present normally. You have copper and iron, for example, in the mitochondria, which will form peroxides. So this is a constant battle. It's not only the ones that are coming from the outside world that would give us problems. And here's this classic Fenton reaction that was described back in the 1890s that shows you how iron, for example, can, that should be iron in the +2 state, will create 2 radicals, leaving iron in the +1 state. And here's a little graphic depiction of all that action happening inside the mitochondria. As I mentioned, outside in the cytoplasm of the cell you have some catalase, but inside the SOD is working to make peroxide, and you've got to have an ongoing way to get rid of these radicals. So hypochlorous acid is also neutralized by glutathione, and in turn will deplete glutathione. This is to remind me to tell you about a patient of mine who had a dental, what do you call those little materials that are put on the front tooth, for example, an onlay? Veneer, thank you. And they used a dental glue. And this lady, within an hour after this was placed, she told the dentist, "My tongue is burning." And he took a look at everything. There wasn't any obvious damage. And over the next several days to several weeks, this tip of her tongue and then progressed through the rest of her tongue and then down into her esophagus. And eventually, she had this jittery burning sensation throughout her whole body. And it turns out she was low glutathione, and after I convinced her to try replacing the glutathione, in this case with the liposomal form we'll talk about, her symptoms cleared up very quickly. And it wasn't until all of this was resolved and we were able to talk about it a little more, it turns out that she'd been using bleach in her bathroom. She had some mold in the bathroom. She couldn't stand it. So every day she'd sit in there scrubbing the mold with bleach, which is hypochlorous acid. And I didn't realize it until I went and read about it, but this is one way to deplete your glutathione, and that's why I mentioned it up here, that it actually can diminish glutathione. And once we got enough glutathione into her, her symptoms stabilized. But she'd gone around, this was months, with this problem, had the veneer removed and seen lots of dentists and was, convinced. And the other thing this illustrates is what's called the chain reaction, chain oxidation. She started out with this reaction right on the tip of her tongue and progressively used up the antioxidant buffers along the membrane, and so it was kind of progressing. It didn't happen immediately, but over days and weeks this was progressing because her system was using up its available antioxidants. And I think it's really a very interesting case exemplifying the slow progression of what happens if you don't have enough antioxidant function. So we're back to talking about glutathione and its interaction with the peroxidase enzyme. And the membranes are made of polyunsaturated fatty acids. And this creates this radical chain reaction that I just described. It's progressive unless you have some material there to stop it. Glutathione, the peroxidase, there's a form of the peroxidase in lipid membranes. And as we'll touch on later, an article has been published just recently using liposomal glutathione, which demonstrates that the cholesterol particle LDL actually has the enzyme glutathione peroxidase embedded in it. So this is going to create an opportunity to understand a number of lipid-related damaging effects, which we'll go into in more detail in the second presentation. So we've talked about the antioxidant function. Let's talk about glutathione a little bit as a detoxifier. I'm not making a call here, I'm checking the time just so I know where I am. Because I may move faster than I realize once I get started here. So what happens? What makes a material a toxin? Almost all the things that we consider toxins are fat soluble. And what does that mean? Does that mean it sits in your belly or wherever you're fat? Well, it actually means that these toxins sit in lipoproteins in the membranes of cells. And what this article showed is that you can take low levels of mercury, non-lethal levels of mercury, and perfuse a cell culture of liver cells with this low level of mercury. And then they went back and broke the cells up into their component parts. And they only found 15% of the mercury in the cell wall. They found 35% of the mercury in the wall of the nucleus, which is kind of worrisome. But look where they found the majority of the mercury. Almost half of it was in the mitochondria. And so what happens when you get this mercury in there, what happens, the reason that mercury becomes toxic is that it creates a massive free radical generation. And it also depletes glutathione. The way glutathione works in our body is to take these toxins like mercury, and the glutathione can bind to it directly, and that takes this fat-soluble molecule and makes it water-soluble. And it has to be water-soluble to both get out of the cell, because the fat-soluble material won't migrate out on its own, and that's why things become a problem. But it can migrate out of the cell, and it can also then be excreted either through the kidneys, although mercury usually goes out through the liver. What's the water-soluble excretion pathway from the liver? Bilirubin. Materials to get into bilirubin have to be water-soluble. And the liver has many defined pathways for moving glutathione in these complexes, either by passive diffusion, and I've also read that they have active energy-requiring mechanisms for the excretion of glutathione complexed with these toxins. This is probably a good point to mention the fact that once this is out of the liver into the bile and out into your intestines, that there is still the potential for problem while there's a pretty reasonable complex there. Particularly if you have inflammation and abnormal flora in the gut, then you can have a chance to reabsorb this mercury. And that's why taking probiotics and having calming down gut inflammation is really important as part of any detoxification protocol. The other thing that's coming to awareness is the fact that some of the cytokine mediators coming out of the gastrointestinal tract will actually block some of the Krebs cycle mechanisms and create a number of problems, including poor energy function, but also even excess weight. Sometimes people they get diverted and make excess fat instead of making energy when these systems are blocked properly. So it has profound implications in our health in general. As I mentioned, glutathione works with a number of enzyme systems. We've talked about the peroxidase, the transferases. Well, the glutamyl transpeptidase is one that will carry glutathione right into cells. This is particularly prominent in the kidney, lung, and gastrointestinal tract. In other words, those cells have the capacity to carry glutathione right into the cell, where most of the time glutathione is broken down into its component parts, imported into the cell, and then reassembled. So to get glutathione into the system at certain times requires an energy-dependent mechanism in many cells. Now we'll touch on why the lung and gastrointestinal tract might need the ability to uptake glutathione, but from what you've seen, you already have a clue. Those are the sites where you have the most oxidative stress happening, both oxygen from the environment in the lung cells and the breakdown of food, which is oxidative metabolism. The other enzyme that's kind of fun to talk about are the glutathione transferases. They're often referred to as glutathione S transferase. And that works, I call it the matchmaker enzyme. You have toxins floating around, you have glutathione molecules floating around. You just have to have enough molecules of glutathione to interact, to increase the chance of interacting. But if your GST transferases are working properly, they will grab onto the toxin and introduce it to the glutathione and increase the likelihood that you'll complex that toxin. The majority of people I see with chronic illness have defects in these glutathione S transferases. Either genetically, we look at those with a gene profile for detoxification, and they either are missing one or both of the genes for these various, there's a GST, mu, and pi, and another one. And almost always in my chronically ill patients, they'll be deficient in one or more of these gene mechanisms. And I think that points out that even a healthy person who's getting along fine, if they're low on these transferases, may run into the situation where you need more glutathione molecules. Someone missing those transferase enzymes, what is normal for one person, they may need a lot more of these glutathiones. Let's see what else glutathione's doing for us. I got introduced to glutathione in about 1995 or 1996 as I was studying mercury. That's about the time that I got involved in mercury detox. And the first thing I did was go and read about why mercury is toxic. And as we've already mentioned, one of the first articles I found said it depleted glutathione. Well, it wasn't until a few years later that I ran across glutathione as being involved in immune function. And it turns out that there's a series of articles that have come out that show that a deficiency of glutathione in the immune cells called macrophages, which also act like the antigen-presenting cells. These are the cells that take an antigen that has, that's what they call things that aren't supposed to be in the body, an immunologically stimulating material, and interact with it. If they're low in glutathione, it will skew to what's been called the Th2 mechanism. Th2 is the types of diseases that are Th2 are chronic inflammation, ranging from allergy to asthma, autoimmune diseases, those types of things. And then Th1 is more of a cell-mediated immunity, which requires a little Th2 but a lot of T-cell function. So the difference between the two is that the cellular immune function is a lot like the street sweeper that's going along. It's going to be great if I lose my voice in the middle of this, but I'll just show you slides if I do. Excuse me. Goes along and picks up debris that's fallen in the street. The Th2 mechanism would occur if a white flour sack fell in the middle of the street. They'd stop the street, block everything off, call in the hazmat team, analyze it, and then have the fire department come in and hose it down. And it would do just what my nose is doing right now. That's you get congested and then you get a runny nose. That's allergy, that's chronic inflammation, as opposed to the Th1, which is that street sweeper action where it just comes in and tears up the material and carts it off for the body to get rid of. And this is, most of the modern diseases are these Th2 diseases, chronic inflammation. So that's what low glutathione will do in our immune system. I'm sure I'll get over this. So bronchial asthma is a typical Th2 disease. This slide talks about the dependency of Th2 balance on glutathione. And it's mediated through the production of IL-12 when you're low glutathione in the APCs, the antigen-presenting cells. So this is a published article that supports that comment. So the primary sources of mercury I don't have to tell you folks about. And thimerosal is one that's found in vaccines. And very recently, a couple of articles from different centers has come out which shows that the antigen-presenting cells, also called dendritic cells, are exquisitely sensitive to this sulfur mercury complex called thimerosal. Everybody knows what thimerosal is. I don't have to go into that. So its mechanism turns out to be mediated. Well, what the thimerosal does is create a Th2 excessive chronic inflammation response. And it turns out that that is mediated by a depletion of glutathione in the immune cells. So there's one mechanism that explains the results of using mercury in vaccines. But any mercury exposure is going to lead to these kind of problems. And when I first got started treating allergy, I was taught and learned that elevations of IgE antibody specific to different pollens and molds is what gave you allergy, and that's going to have a genetic source of problem. But I see people all the time who keep coming to me and say, well, I was fine, I didn't have any allergy or runny nose problems until 2 or 3 years ago, and I'm now getting worse and worse. And I used to think that was just they were beginning to express this problem that had not been uncovered before. But now when I see somebody like this, I think about, well, how much mercury have they been exposed to, and what's their accumulation? And it really turns out to be quite true. Let me clear my throat as well. Is anybody sensitive to mold in here? Moldy environments still get me, although I'm a lot better than I used to be. How's that? Am I back on? Okay. So it's the depletion of glutathione in these dendritic cells that predisposes to this increase in chronic inflammation. And it turns out that one dendritic cell is going to interact with 300 T cells. And these T cells are going to carry the information further down the line of whether to go to this fire alarm, four-alarm, fire alarm type problem, or to use the street sweeper mechanism called Th1. And that's how this whole mechanism gets perpetuated, and it goes on and on. So it takes a while once people get depleted of mercury to improve, and it goes a lot faster if you have a way to build their glutathione. It also turns out that not only does mercury do that, but the endocrine disruptors, the things that you end up in with plastics and things like that, they also mediate a Th2 polarization, and they do so also by depleting glutathione. So you begin to see how this pattern is being repeated over and over. And here's the article that shows that they can have increased serum IgE, which goes along with explaining why the incidence of allergy is increasing on a continual basis. I have an interesting patient that I saw who was having, he's an 11-year-old with Epstein-Barr virus, so-called fatigue syndrome, related to EBV virus, they thought. And he'd been sitting at home for almost 6 months when he came to see me. And in the course of his evaluation, he did have allergies that had been progressing for 2 or 3 years where he had no symptoms before. And using liposomal glutathione preparation, his symptoms improved a great deal. But his fatigue persisted and his viral titers persisted. And in the course of his evaluation, we found that he was loaded with mercury, and put him through a mercury detox protocol, which took several months to completely affect. I use intravenous infusions of nutrients as well as DMPS, as well as oral chelators once they get stable. But the point is that once we got his mercury level down, all of a sudden his spleen went back to normal, and 6 months later he was able to return to school and to playing soccer and cross-country in the same season, which is really quite remarkable for somebody who had been incapacitated to the degree he had. And I think I've not had many cases where it's been that clear, because his EBV had been worked up continually over the preceding months. His mom was, his parents were besides themselves trying to figure out why this kid was sick. And it just turned out by coincidence that we were able to find the mercury. Of course, I've learned to look for it, but I don't think makes me wonder how many people are running around with these chronic inflammatory mechanisms that are stuck because they have an excess of toxins. And the point here is that you really couldn't get ahead of the problem just by replacing his glutathione. He had such a drain on his antioxidant system. So here's a article talking about the role of mercury in autoimmune disease, which we've already mentioned. It's really just a dysregulation of the immune system. And the cells, they're now defining a whole new category of T cells called Treg cells. And these Treg cells are actually very sensitive to mercury also. And it creates just the kind of confusion in the environment of the body in which you can get autoimmune disease, or you can let some of these intracellular pathogens creep into the body, that, like viruses. And then more recently we're seeing a big surge of concern about diseases like Lyme disease, which again is another intracellular parasite-like material. So this goes into the mechanisms and also what happens with low glutathione can actually lead to apoptosis in microglia and astrocytes. Well, microglia are really macrophage cells that are located in the brain. So it shows you the impact in another mechanism. These two things, depleting, increased mercury, depleting glutathione, starts to cycle and create all kinds of mechanisms in the neurological system also. So this points out that just taking 3 thimerosal-containing influenza vaccines can raise the exposure quite above what you would like to have. And I think you're going to get more detail on thimerosal, so I'm not going to go into that any further. So what about mercury and other diseases? This fellow Joachim Mutter over in Germany has written an article in 2005 in which he relates the increase in autism over the past decades, paralleling the cumulative effect of mercury exposure, and the finding that autistic children perhaps exposed to mercury during pregnancy may be predisposed to having problems later when they get an additional mercury insult. And this lady, he found also that mercury could inhibit an enzyme that we're going to come to in just a minute that's involved in the formation of glutathione. And what this is the methionine cycle. So we're going to spend a couple minutes running over the biochemistry of this, because I think this is really important. You don't have to remember every step here, but if you just have this image in your mind of this cycle working all the time in the body to create the components that go on to form glutathione, this is going to turn up in a lot of diseases. I mean, yes, it's autism in kids because these kids' brains are still developing, but this shows up, and I'll illustrate a point at the end about a case, what happens when the cycle gets blocked. Well, the methylation, that sounds like a big word, but it's just a single carbon being moved around through this cycle. That's what the whole thing is about, the movement of this carbon to different components of this cycle. And the cycle is primarily methionine going to homocysteine, and there's some intermediates called S-adenosine methionine. You've heard of that as SAMe, and S-adenosine homocysteine. And then the cysteine can be broken down to, the homocysteine can be broken down to give us cysteine for glutathione, as we saw earlier. But this cycle, the key to understand this is this cycle has to go around on a continual basis to form cysteine. It needs 1.5 cycles to go around in women to make cysteine. And this lady whose article, Jill James, I'm going to quote you in a minute, says that in boys it has to go around twice. And she thinks that's one reason why young males are more susceptible to autism than females. But the whole point here is that a blockage does not necessarily cause the next thing up the line to balloon up. It kind of stops the cycle. And that's the intriguing aspect of this. Now, those of you who have read about elevated homocysteine, there's been a lot of articles trying to make that a toxin material. And it doesn't turn out to be quite as toxic. It is in some ways by itself. But really what it's probably an illustration of is a biomarker of this cycle stopping. And it is related to a deficiency of B12 or a deficiency of folic acid. will also cause a buildup in homocysteine. Well, it turns out in the kids with autism, they actually had low homocysteine, and they were piling up a bit in the S-adenosyl homocysteine. That was elevated. But they have low cysteine and low glutathione, and giving them plain B12 didn't do anything. They gave them plain B12 and various forms of folic acid and trimethylglycine, which is another pathway that will help move this cycle, keep this cycle moving, and nothing happened until they added methyl B12 or methylcobalamin. And then all of a sudden they began to see the end products, the cysteine and the glutathione, rising up again. It didn't cure the problem, but here's the intriguing thing about all this. In order to make methylcobalamin, you have to stick a carbon on it. And to stick a carbon on methyl B12, there's an intermediate step called glutathionyl cobalamin. So the irony in all this, and this is published, I didn't give you the reference, but is that you need a glutathione molecule to pick up the carbon to hand off to the B12. It will pick it up from folic acid. And so if you're not making glutathione at some point, this cycle won't work. And you'll end up with stopping that whole methionine cycle. So while methyl B12 is the key to the methylene cycle, it takes glutathione to keep that working. And so ultimately, this is where people get into this chronic illness spiral, chronic fatigue, these kind of problems where these kids are stuck and they don't come out of these difficult problems, I believe, is because they no longer can methylate. For this methionine cycle, it turns out that coming off the cycle, SAMe goes on to make the myelin and so in the brain, and so there's a whole bunch of interrelated mechanisms that come off of the methylation cycle. And that's why sometimes when you give glutathione to children like this, some of them will show an improvement in their function. We used to see that first with intravenous glutathione, and now with the liposomal glutathione, although I don't want you walking out of here thinking that's a cure, because most of these kids have then, by the time it's apparent, they have deficiencies in their myelin, they may have oxidative stress, they may have autoimmune antibodies affecting the brain, they have chronic inflammation, they have GI tract problems, and all of these things need to be addressed in order to solve the problem, if you will, or allow them to function. But glutathione plays a very key role there, and the metals do too. It turns out that because of the lack of glutathione, they are accumulating metals. I don't know which is the chicken and which is the egg necessarily, but you'll almost always find metals in those kids. So in my opinion, during states of excess oxidation, an outside source of glutathione is needed. If you remember that Mutter article, this is where methionine synthase works, is in using the methyl B12 to form methionine, and that's susceptible to oxidative stress and is blocked by mercury. It turns out that this enzyme can be blocked by mercury, and this is what we found, and the reason I know this is because this child's with the EBV that I mentioned in mercury, he had a low glutathione in his blood and a high excretion of taurine. I apologize I didn't give you the taurine part of this. The cysteine can form the amino acid taurine also, and that's a great antioxidant, but it doesn't work with the enzyme systems, even though the liver likes it. It's kind of a fallback, if you will. And so he had an elevated taurine. It was about 3 times normal, and a very low glutathione. And then we got the mercury out, and we supported his glutathione with liposomal glutathione. These went from abnormal back to normal, and that's when he was able to go back to school and back to function. So here's Jill James's article, or the reference. Her article came out in December 2004. If you want to look, you just Google or use your search engine for Jill James and autism and mercury or methylation, it'll come right up. And so the whole point that James made at this presentation later was that the finding of low glutathione is that children of autism will be less able to detoxify and eliminate these heavy metals, and that obviously contributes to the continuation of the problem. So it turns out mercury is going to be the great equalizer in this world. The New York Health Department has found mercury levels that are 3 times those that are found in the rest of the U.S. They're highest amongst people that tend to eat sushi. And they also came out and said that these levels are not worrisome. You know, I'm telling you, it's like, you know, accumulating mercury is like running uphill with a backpack full of rocks. You know, you're going to get tired out eventually. And maybe you found along the way that if you could take a few of those rocks out of the backpack, you can move uphill a little further, a little longer. But mercury, I think, is one of the biggest problems that we face. And its release continue. One reason everybody yes, the ones that eat fish have more of a problem, but coal-fired generation plants releasing mercury into the atmosphere is blown towards the north and east from the Midwest. Yeah.
Back in that cycle where the glutathione for the methylation,
Yeah.
is that just a drop-off, the glutathione stays available, versus when you use the glutathione to detox the mercury, do you lose the glutathione?
Yes, when you detox, Boyd Haley will tell you for every molecule of mercury that you remove from the body with your natural mechanisms, you lose 2 glutathiones, actually 1 going out with the mercury molecule, but you lose 1 that goes into the oxidized state because you have to move mercury from +2 to +1 to make it available for the glutathione. So you're really putting a big demand on your glutathione system. And everything works fine until it doesn't, and then you get sick, and that's why you get, you know, and that's why I'm telling you, you look at, you know, somebody they're doing fine and then they're starting to get allergies and they're starting to get lethargy and they're having, you know, viruses that are occurring more often. That's the kind of progression that I see, and I often will find mercury in those people. They'll stabilize by boosting their glutathione in the interim, maybe with N-acetylcysteine or whey protein, and I like the liposomal glutathione. There's another question in the front.
In the Krebs cycle, when you're having bivalent metals being used as enzymes to take it step by step in production of ATP, does the mercury being a bivalent metal prostitute the trace minerals that are being used? Is that one of the mechanisms of it?
I'm sure
it makes that enzyme not operative?
I think that's exactly how mercury works. It turns out, I can't give you the references because I've seen this from one of the labs, and someone else has corroborated this, but I don't have the reference yet. The first 3 cycles in the Krebs cycle, the citrate diisocitrate, is dependent on having glutathione available. So you're going to have mercury both displacing the normal minerals that are used for that function, as well as pulling the glutathione out of there so that they're not available. And that happens in that porphyrin cycle also. You know, while the specific metals seem to have an impact at certain sites in the porphyrin cycle, I think that some of these are also dependent on glutathione. And so that depletion can have a broader spectrum, but you're absolutely right, and that's where, yeah, several places in the Krebs cycle. So 25% of the people in New York City have elevated mercury, but you know, even in the Bay Area, I live in the San Francisco Bay Area, and you know, there's warnings all the time not to eat fish out of the bay, and there's articles in both the New York Times and also the San Francisco Chronicle on a regular basis, and you can find these in their search engines easily, talking about mercury. But don't worry, it's not a problem. According to the newspaper article in the health department, I think it's personally a big problem. 10 to 25% of women have elevated mercury. Nationally, 10% have 5 micrograms per liter or more, or less. It used to be the industrial level at 13 micrograms per ml. If you got, you know, they do blood testing at the end of a shift, at the end of a week, and if you had that much mercury in your blood, they wouldn't let you work in a mercury plant, you know, a plant using mercury anymore. So half, you know, half of that, or almost half of that, at 5 micrograms is really kind of interesting. The more fish you eat, the higher it is, but the implication is really profound. If all these moms are going around, you know, I mentioned Dr. Muter's study, if all these people are going around with elevated mercury in their bodies, what is this doing to the kids? And there is now just published some work that suggests that in mice, that if the mother mouse has a lot of mercury, that the kids may not make glutathione peroxidase properly, and so they may be even more susceptible. And that really does create an interesting linkage in the chain of events towards understanding some of the problems with autism, for example. And exposures early in life, according to this article in the Denver Post from January 28, 2008, says that exposures to mercury early in life may cause problems later. And there's growing evidence to that effect. And this was based on monkeys that had been given mercury young in their life, and then like a decade or more later, they were evaluated for their brain content and brain function, and the ones with mercury exposure didn't function as well. So I wonder if we're beginning to see all this in our society. And this talks about the delayed appearance of toxins exposures. Some of the things they thought were due to normal aging might in fact be due to the ubiquitous environmental exposures like lead, said the doctor from Johns Hopkins. The fact that it's happening with lead is the first proof of principle that it's possible. Pollutants like mercury and pesticides may do the same thing. I believe this was the animals given the metal. So we now also know that exposure to pesticides raises the risk of getting neurologic disease like Parkinson's, even a decade or more later. Well, we've talked about the coal-fired generation plants. This was in the New York Times, April 26, 2008. No, couldn't be. 2007, my apologies. I'm just ahead of my time, right? And diesel exhaust produces a compromise in those cells that we talked about, the immune cells, that goes on to create allergy and chronic inflammation. And that's been documented. It also turns out that that's mediated by oxidative stress and reversed by increasing their glutathione. And there's your article, Journal of Immunology, 2005. I can give you the PMID on these if you want. That's the, the PubMed ID number. It's easy to search that way. Just if you want it, just raise your hand. I'll read it off if you can't read it. This has been known for some time about the, it's not accepted, but that you can take rabbits and expose them to ryegrass and they won't get allergic unless you expose them to diesel exhaust first. That's where I first learned about it, probably in the late 90s. So diesel fumes, is this why there's a rising incidence of atopy? That's allergy disease. And so this article in Journal of Immunology questioned whether this was an urban legend. And they indeed showed that treatment of human B cells with polyaromatic hydrocarbons, that's what's released in the exhaust, creates a major constituent of the problem that induces the production of IgE and enhances the production of IgE. It's the same mechanism. They're getting their antigen-presenting cells depleted. So here's a list of toxins that require glutathione for removal. Now we've got a documented list that includes mercury, diesel exhaust, pesticides, endocrine disruptors like PCBs, DDT, and the non-polychlorinated, whatever they call that type of PCB. So it should hardly be any wonder that we're seeing a rise in some of the types of problems that we're seeing, including autism, for example. So when the researchers say, well, it doesn't appear to have anything to do with the mercury in the vaccine, there may be other factors that are contributing to this and compromising both the parent and the child. And man, I got through that.